Good morning, everyone, for joining in today's CPHMS webinar, Center for Predictive Human Model Systems webinar. Since past 1 month, we've been having, although we've been having this webinar series for close to 2 years now, but since last month, we've been having a new series where we have been focusing on computational and predictive modeling in biological systems. And this time, we are pleased to have Dr. Shandar Ahmed, who will be talking about
Surat, I think your full face is not coming. You're too close to the camera, maybe, or you have to turn it.
This is
your half of you is getting cropped up. Not yet. I think you move your camera a little upwards, so your face will become fully visible. Yeah, because you're recording also. I thought it would.
Sure.
Yeah. It looks nice.
Can I also request everyone to keep their audio and video is okay, but audio mute during the duration of webinar. So before I introduce Dr. Shandar and we start with the webinar, I'll just take briefly a few minutes of your time to talk about our center and why we have been having this webinar series for quite some time now. So I'll just share my screen. Hopefully my screen should be visible. So yeah, so one second. So I am Surat and I'm a senior research associate at the Center for Predictive Human Model Systems. And this center was established almost, one second. Okay. Yeah, so the center was established almost 3 years ago now to as a science and policy center to enable systems that are more relevant to human biology rather than primarily relying on animal models. Okay, so what do I mean by human relevant model system? So while, like I said, while biology has primarily relied on animal models for a long time, in the last few decades, we've also developed several systems such as organoids, organ-on-chips, 3D tissues, advanced imaging techniques, which has also helped to generate data that is more relevant to human biology. And also in silico models that are built using this human relevant data can help us not just capture, but also predict the subtleties of human biology, which may not be possible using animal models. So towards this end, our center has been involved in several activities, including increasing awareness in this space, because in India, especially, the awareness for these new model systems is currently lacking. So towards this, we have published several popular articles, reviews, and webinars. And we've also been involved in education and training because we would also like to create a skilled pool of researchers who are trained in these methodologies. So we have conducted again several workshops, and this year we'll also be conducting the first India EMBO lecture series in the area of microphysiological systems towards the end of the year, and we'll be coming out with the details for that very soon. We've also involved in several policy initiatives where we have written white papers, conducted multi-stakeholder roundtable meetings with government and regulatory bodies to provide recommendations in terms of how we can further enable this field in India. We're also involved in several global collaborations to increase the knowledge base of these methods, both in India and globally. And we've also been involved in funding where we have provided grants to 2 Indian scientists to develop an adverse outcome pathway in the field of cancer. The roadmap that we envision to kind of enable this field in India is, one, we would like to work towards establishing dedicated centers of excellence to further develop and validate these methods. Another aspect is connecting the technology developers with the end users, who may be in pharma companies, etc., and to further facilitate the technology transfer. We would also like to engage with the regulatory bodies during the development of these technology, during the initial stages of the technology development, so that the ultimate point of the regulatory acceptance, when we reach that point, it becomes a much smoother process. And many of these technologies are much developed across the globe. So we are also trying to enable more cross-border collaboration for both technology transfer and international funding. And in the end, one of the critical things in India is, especially for microphysiological systems, we would like to enable the supply chain for indigenous development of this technology, which could further bring down the cost of working in these methodologies to a great extent. So with this, I would like to stop, but I would also like to say that we have a quarterly newsletter. I'll put the link, the registration link in the chat box, where every 3 months we bring out a newsletter which covers all the latest articles, op-eds, and we also have informational videos which cover the recent happenings in this field. And with this, I'll stop. One second. And just give me 1 second, and I'll introduce our today's speaker. So today we have Dr. Shandar Ahmed, who is a professor at School of Computational and Integrative Sciences, JNU. His lab is interested in developing data-driven algorithms and applications for biological data with an integrative perspective. And their technical interests lie in machine learning, big data analytics, novel architectures, training methods for deep learning through neural networks. And now, over to you, Dr. Shandar, for having today's webinar. Thanks a lot.
Thank you very much. And thank you for keeping it brief for the introduction part. Yeah, so I'll share my slides first, directly going to the point. Can you see the full screen slides, or you're only seeing?
Yes, I can see you.
Is going full screen to you? Or?
Yeah.
Okay.
Yes.
Fine. So thank you very much. And your Center for Predictive, I can't remember the full terminology, but it's a wonderful center. And for during last 3 years, you have done activities, and it is such a privilege to be associated with it. Its activities in the past and today. And thank you for connecting me again. And today, as the topic stands, tools for predictive modeling of biological systems. I'll not be listing out a list of tools here. We'll basically take a philosophical view, because this center, the science policy predictive systems. So can you hear me now? I think I was muted by mistake.
Yes.
Okay. So since predictive modeling of systems, human model systems and other systems is one of the primary focus, so we'll take a look at what we predict and what we mean actually by prediction. And while developing novel tools for prediction of biological systems, what care has to be taken, and when we look at a tool, what is to be seen in this tool to basically believe the tool and to trust the outcomes that these tools produce. So from that perspective, I'm not talking of a list of tools. Of course, there are agencies which are collecting a list of tools based on the kind of problems you are addressing, and that data is available. And here I take a different, slightly different perspective. First of all, and then I'll take a case study of our own systems that we have been studying. We have been studying the protein-DNA interactions as one of the primary objectives of our lab. Other than that, we are also studying medical data such as ECG and MRI and all those things, and the drug design perspective. So 3 perspectives we study in our lab. One of them I'll take as a case study, and how we have developed tools to study protein-DNA interactions and how they have helped us in terms of the predictions and in terms of understanding the biology, the basic science, and also applying them to solve the problems of application nature. So first of all, why do we make predictions? We make predictions all the time. You may have heard of predictions from the astrologers, predictions from everybody around us. They make predictions. For instance, somebody will say tomorrow there will be rain. They may have had no particular idea about why they are saying that there will be rain today, but they have some ideas. They sometimes make predictions about totally unexpected things and unexpected outcomes. People, there are people around you who would make predictions about things which science cannot predict. So what are these predictions, and why do we make predictions? And there are so many prediction methods or prediction approaches that are prevalent in our society as at large. So here I have put together kind of approaches that people have used over long period of human history to make predictions. As we say that we want to know about the future. We want to know about the outcome of our actions. We want to take a step. We want to make a prediction whether this step will work in the direction we want to work or not. That is how we do it every day. Our everyday life of an individual and a society is based on certain projections and predictions, and then the quality of outcomes depends on the quality of decisions we make to achieve the same outcomes in a predictable manner. So if our predictions are good, if our projections are more reliable, we lead a more a better life. We may made the quality of our life improves, and many of our hardships can be removed in this process. So to take appropriate decisions at an individual level, at the society level, or at a molecular level is pretty critical for our understanding and dealing with nature. So if I can take the liberty of dividing the kind of predictions that are all around into 4 categories, one of them anecdotal predictions, which all of us do, and some authoritative predictions, and data-driven predictions that we are primarily interested in here, and the first principle predictions. I'll quickly take a look at what these 4 types of predictions mean for us. So for example, anecdotal fallacy has been talked about by fallacy in logic. So the anecdotal predictions work in this way. Somebody may define, first the definition of anecdotal prediction or fallacy is that people use anecdotal evidence, isolated examples that rely on personal testimonies to support or refute a claim. When you look around you, a lot of people will give you logic of the kind that has been shown in the right example on this slide, which says, my grandfather was a heavy smoker most of his life, but he lived to be 90 years old. Therefore, the conclusion is that smoking is not harmful to people. So this is an example we hear from a lot of people all around us, and all of us make these kind of casual predictions and often contradictory predictions. So this, it is not just a domain of common men. It is not just in the parlance of common men. I have seen doctors saying that this particular medicine, although there is no evidence, there is no report that this medicine works this way or this dose works this way, but there is anecdotal evidence. That means the doctor has seen encountered certain experiences in his clinic, and based on that clinical experience or isolated examples, he may have made certain anecdotal conclusion or a hypothesis, which is based on which he makes certain predictions. Whether they are correct or not, that is not my point right now, but what I'm saying is that this is how some of the predictions are actually made. Another is the authoritative predictions, which are primarily faith-based prophecies. Like you would go, they will tell us that this world will collapse on that particular day, and if you take this route to this way, this will happen. So predictions based on primarily faith, which has been passed on from one individual to the other or from one generation to the other through the process of faith. So this is usually passed on through a cascade of followers. You believe them or not, that is a separate matter, but this is an approach to make prophecies. No questions asked. This is primarily a matter of belief, and they have been declared in the form of authority of certain spiritual power. On the other hand, the data-driven predictions that we talk of today are different from these two. They are, within them, I've categorized 2 types of predictions. And I'm using this I'm taking liberty of defining these 2 types of predictions as data-obsessed predictions and data-conscious predictions. Data-obsessed predictions are those which can detect patterns which do not exist, and which will make fit an existing data between the features of your data and the outcomes of your data. They will make wonderful assumptions, and they will say, okay, my model is 100% correct or 90% correct. And the patterns that did not exist, like if you can recall that movie, Beautiful Mind, that the Beautiful Mind was able to see patterns in numbers, in newspapers, these patterns like a hidden code. So these code did not exist, but your mind is predisposed to seeing these patterns, and therefore non-existing patterns are sometimes discovered by people who are data obsessed. And the same way data obsession can occur. This is about the kind of unsupervised data obsession, and the supervised data obsession would be to produce overfitted models which give you a very high accuracy while you're developing these models, but they have no practical value in the real life, because when a new situation is encountered, new data comes, these models do not work. So such kind of predictions can be called data-obsessed predictions. When you are simply obsessed with your data, you are simply obsessed with your patterns, and you want to discover something which did not exist. So for instance, if you have data and you want to do a clustering, every clustering method will produce some clusters. Well, there are methods to cross-check whether the clusters that you're now getting are really meaningful or not. But if you keep on tweaking with your parameters, you're tweaking number of things, and at some point of time, you will get to see some clustering in the data, which is simply an outcome of multiple hypothesis testing. So if you keep on trying and you do not even report how many different things you have tried to reach this conclusion, so many a times these non-existing clusters will appear to you, and overfitted model will try to show you very high degree of accuracy. In both cases, they have no practical value. So the solution to the data obsession is the data consciousness, which can be the predictions based on this approach can be considered to be data conscious predictions, where the important things are to understand not just the data, but also the context of the data in which this data was collected. And also not just looking at the data in front of you, but also looking at the knowledge on that particular domain, on that particular subject that you're investigating. What is the state of knowledge? What do people know about that data, about that particular biological problem? So that's why a new science of artificial intelligence is now emerging, which is called explainable AI, or in the context of data science, we can call it explainable data science. So the models that you generate, the patterns that you discover, are they explainable? Are they consistent with the knowledge in the domain of the subject that you're investigating? So these are the considerations when we make predictions. And finally, there is another type of predictions, which is called the first principle predictions, which are based on primarily the laws of nature, laws of physics. And these type of predictions can be called ab initio. When you start looking, if you are looking at a molecular system, you start looking for the atomic forces, you collect you model every individual force, you create mathematical model. But the irony in this case, the physics-based predictions, is that the first principles that you have generated were originally data-driven. So there's no escape from data, actually. So the first principles are data-driven. Secondly, the application of the first principles is so complex and so complicated, the large systems cannot be treated with the first principles. But definitely, if you can somehow find the first principles to solve a problem, you should not be using a data science approach to this. The need and the challenge is that the first principles, in most cases, although the first principle of the Coulombian forces, the Newton's laws, even the fundamental forces of nature are already known, but our real systems are so large, so huge, and so complex that they're simply scaling up those first principles to be able to address a real problem are really almost impossible to achieve. And in the current scenario, as of now, we do not see any major biological problem being solved with good application from the first principles without the help of data science. So that is why the data science and the predictive science becomes important. The first principle predictions may not even be called predictions. They may be called simply first principle calculations, ab initio calculations, or de novo calculations. So now if you put these two together, one against the other, you can call them deductive approach, which is basically from the first principle, you deduce and you predict what will happen under given conditions. And the inductive approach is the data-driven approach. When you have the data and you know that in these thousands of examples, this is what has happened, then from this, out of these thousands of examples, you build a narrative, the computational and mathematical narrative, which will link the observables to the non-observables, unobserved features, and then you induce or you induct a new hypothesis, or based on that, you make a prediction. So in the case of biological systems, all these things apply, and the purpose of predictions in the biological systems is, of course, biological in nature, and the first of them would be to predict and prevent diverse events. So for instance, if I have the records of ECG, can I train my machine learning or deep learning models to read a normal ECG, apparently normal ECG, which doctor is not able to read, and can I tell in advance, 1 hour before or 2 hours before the heart attack, that this person is going to have a heart attack, or this person is going to have a hemodynamic shock, as I was discussing yesterday in a PhD viva. So can I make predictions 6 hours before, 10 hours before a particular adverse event happening? If we can do that prediction based on not the actual symptoms of the disease, but based on the maybe related or unrelated activities such as ECG, or maybe something that I'm constantly monitoring, or the heart rate or anything, rhythm. If based on that, I can predict it 6 hours ahead, I can prevent these diverse events. So that is one purpose that we do predictions in biological systems. Another thing, another context in which we look at the impact, impact of an intervention. When I'm trying to design a drug and I want to treat a particular disease. So, for instance, suppose I already know the target to which I want to activate, or I know I know the target for which I want to develop an inhibitor. So if I know that this target will perhaps treat this disease, this is the mechanism of disease, or so I have a candidate drugs, and maybe my candidate drugs are in millions, or maybe if I have already filtered them out, maybe in thousands. So I cannot take all those 1,000 drugs to the clinic and I want to know which particular drug will work. So here I want to make a prediction about out of these thousands of drugs, which will have the desired impact. So in order to see the desired impact, I need to make a prediction about what this drug will do. And that is how in this scenario, I have to model my molecular machinery in a computational context. I can, because the real life experiment is neither desirable nor possible. So this is the second scenario in which our predictions are aimed at looking at the impact of an intervention. And third is also this is more like a basic science. We create a hypothesis and we use prediction. So if I say, for instance, if I want to ask whether the structure of a protein depends on the sequence, or whether a particular interaction with another protein or DNA depends on the sequence or depends on some other chemical or solvents. So what I can do is I can collect a lot of data and I can try to model the inputs of this, let us say, sequence features with the actual interaction. And if I am able to create an accurate model, that means my outcome of this property is predictable from the features I'm investigating. And if my outcome is predictable from the features I'm investigating, there must be some relationship. Otherwise, they would not be predictable. In the modern day deep learning and in modern day data science, some of the features that actually make the predictions may not have an obvious or direct relationship, may not have a visible relation with what we are trying to predict. So as I sometimes said that if in the past, if you ask a question, can I predict the health of a person from the way he walks? Can I tell about the heart disease by looking into the eyes of a person? So these apparently look unrelated, and these are not valid questions to the biologist, say until 10 years ago or 15 years ago. But today these questions are valid, and the answers to them are coming up. So there may be very indirect relationship, very indirect association. There may be common factor which is affecting the two unrelated diseases, and therefore you may come across a situation when totally unrelated features make a prediction about something which none of us had seen. So asking these kind of questions to deep learning, to machine learning or data-driven sciences has become totally acceptable in the age of data science. Because our goal is not to look at the first principle, the mechanistic basis of what is happening, but in these cases. But then if you keep on doing this predictability, pretty much everything can be predicted with everything else. And if you are data obsessed, means if you are data narrow-minded, if you are data fanatic, means you believe the data that you have, and you are so narrow-minded about your data that you are ignorant about what the data that exists elsewhere. So if that is your approach, you will end up with wrong models. You will feel that, yes, this disease can be predicted from these observations or these measurements, but actually it is not. It is just because the kind of data that you're dealing in that data, that kind of relationship existed. So in this, you have to juxtapose three different things about the predictive models. One is the prediction itself, that we make prediction, we made machine learning models, we detect patterns, we make predictions. And the second debate that you often come across is the causation versus correlation. Two properties may be related to each other. And for instance, there may be a particular ethnicity in which a particular disease is more prevalent. So can you attribute this to some genetic factors? Not necessarily. It may be the social circumstances in which that particular ethnicity has been placed, which is causing that problem. So that may have nothing to do with genetics, nothing to do with ethnicity. So these kind of correlation, many times we observe these correlations. And if you develop a predictive model in such a scenario, when you find the correspondence between, say, ethnicity or between the physical features of an individual based on the skin color, based on the height, based on the body mass index. If you collect these profiles and you make up predictive systems and you say, okay, and your model comes out with a relationship that if I take the ethnicity, the income, the dress code or makeup, and I relate it, my predictive model may say, yes, these things determine whether a person will live long or not, or whether the person is honest or not. So these kind of totally absurd correlations might exist, and totally absurd outcomes can be produced if we inappropriately used our predictive systems. So on the one hand, totally unrelated properties can be related with the help of predictive modeling. On the other hand, totally absurd solutions or totally absurd hypotheses can be generated by the same methods that are producing something useful for us. So here is where we have to pay attention to what kind of predictions we are making, what kind of hypotheses we are making. Are we data obsessed or are we data conscious? So data obsessed here, in the context I'm using is the data that I have. So if I'm obsessed with just this data and I'm ignoring every other data in the universe, that I'm data obsessed, I'm data fanatic. And secondly, that this is one thing that I mentioned. And then the second thing is, if you find some correlation, it does not indicate a causation, because two things may be related to each other by many other factors. It may be that both of these factors are governed by something common, or maybe just it is a coincidental one. So coincidental correlation may happen and causative correlation may happen. So the correlations may not be an indication of cause, because the two properties that you are trying to investigate are actually not related directly. It is, secondly, this may be by chance. If you are trying to try too many things, if you have collected millions of features of a data and just a few thousands of data, many absurd relationship will exist, because the biological observables do not move or do not act in such a systematic way. Every biological observable is impacted by so many other observables. So these observables that you find in your data may represent a totally chance-based correlation, and this chance-based correlation is happening because you're not taking full care of the data science approaches, appropriate robust data science approaches, such as the multiple hypothesis corrections, such as the overfitting of the data. So these things become important for the prediction scientist, for the scientists working in the predictive modeling, to take into account the kind of data they are looking at, the context in which the data was generated, and so on. So sharing it further, the tools for the predictive modeling of biological systems can therefore be grouped into two categories: the mathematical and physical tools, which are based on mathematics, and the statistical and data-driven tools that I have just talked about. And primarily, the statistical and data-driven tools are machine learning and deep learning-based models. And what is, now you would be wondering that if everything can be solved by mathematics, by the data, then what is the place for biology? We are talking of biological systems, and we are not talking at all about the biology in it. So is there a place for biology in the modeling of biological systems, or we can simply take data and do the modeling and try to and claim that we have solved the problem? So here's the answer. The biology of the biological modeling appears and is crucial for, number one, generating and collecting the data. At the time you collect the data, are you taking precaution of controlling the biological conditions? Are we taking the biological replicas properly? Are we considering that if I'm studying a particular tissue, I'm taking the sample from the right place or not? I'm taking the right size of the sample. Am I ensuring that it is not affected by the biological conditions? So that is where the biology of the biological modeling will come. Second is biology of integration will come in the form of data integration and domain-specific fine-tuning. Many a times you will see that the data appears in our literature or in our databases by different names. Same entity is named X in one database and Y in another database. So how to map X to Y? A data scientist cannot do it without the domain-specific knowledge. Secondly, even if the data scientist knows that this X corresponds to Y, the degree of granularity, the detail at which the X has been defined and the details at which Y has been defined is not straightforward. Many a times we come across this. If, for instance, people who may have analyzed microarray data, they would know that at probe level, the gene expression looks different, and the gene level, it looks different. And even the gene names are not identical in different databases. So how to, or the tissue and cell. So people have collected data from cell and at the tissue. People may have written something like the expression coming from skin. Some may have talked about epithelial cells. So some may have talked about other layers. So the details in which the different data sets are collected. And if you want to integrate those, since I said you do not have to be data obsessed, you don't have to be data fanatic. If you don't want to be data fanatic, that means the problem that you are investigating, the data that you have, has to be put in context with the data that exists elsewhere. Now, when you start looking at the data elsewhere, you find that this data is so complicated to put in context. You had collected the data at the gene level, or you had collected the data at the isoform level, but the data available in a database is not at the isoform level. They do not have the SNP information. You had the images of resolution 360 by 360. There the data was at a higher resolution or at a poor resolution. So this kind of data integration problem is to be addressed at the computational level, but also at the biological level, where you understand the exact conditions and exact context in which the data was generated and produced, and integrate this appropriately. And then the third is the feature engineering. That is when I want to achieve something. As I said, I want to find the impact of a drug. So if I want to find the impact of the drug, I want to predict the impact of the drug, then I have to characterize my drug. I have to characterize my target. So how do I characterize it? What features are important and what features are not important? So these features will not automatically come to a data scientist. These features have to be provided by a biochemist or a biochemist. So biological modeling of features is therefore critical to this. And then finally, what is the hypothesis? What do you want to prove? So do you want to say that this drug will kill the cell? Or do you want to say that this cell will activate a particular pathway? Or do you want to say something different? So this exact definition of the hypothesis is also the task of the biological expert or the domain expert. So finally, as I said in my previous slide, that we are in the age of interpretable, explainable artificial intelligence. So a few years ago, we were all obsessed about data science, artificial intelligence, machine learning. And now we are graduating, we are realizing that AI is trying to act too smart, and is trying to give us too many answers which are not actually working. So we are now trying to incorporate the domain knowledge or the knowledge from the biological context of the problem, so that we have the AI which is interpretable and which is explainable. If I find a solution, I should also tell why this solution is appearing. So in some sense, this is an integration of the inductive approach and the deductive approach. So finally, I'll take a quick look at the kind of system that we have been investigating, that is protein-DNA interactions. So can the host tell me how much time I'm left? So I'll go through the remaining part accordingly.
You have some time. You can take 10-15 minutes.
Okay. So I'll look at the protein-DNA interactions problem that our lab has been investigating for a long time. And the other problems I'll not touch upon, because I take just one case study so that I can explain the point of overfitting, the point of the increasing complexity in systems, and how we can, when we develop tools, what are the considerations that we should have. So if we just look at, for those who may not be so familiar with the biological systems, that the protein-DNA interactions are critical for many different lives events, such as gene expression, the sensing of pathogens in the innate immune system, the genomic DNA packaging, and the regulation. So in these, if I skip this, so first a question that we have been interested in to know, can I annotate all the DNA binding proteins in the genome? So for that, we have to answer the question that what kind of proteins bind DNA. So again, this can be done with a predictive modeling. If I know that the properties required to bind DNA are such and such properties, then I can simply collect those properties. These properties should be computable from the protein sequence or protein structure, whatever we know about. And then from these properties, I simply have to develop a classification model, and my outcome will be DNA binding protein or not a protein. So in the computational language, it will be class level, positive class level, and negative class level. So how do I present it? So several years ago, we started this question, and we started looking at protein structures, and we found that these three properties of the protein structure, which are easy to derive, such as the electrical moments of the dipole moment, the total charge, and the quadrupole moment, can produce a very good model. So this example is important in the sense that if we are looking at a relationship of features that we know, the properties of proteins that we know, and the property that we do not know on the one hand. So what is the relationship exists? Should we have lots of features, or should we intuitively think of some simpler features? In this model, when people at that time were working on topologies of the patches and charge distributions, geometries, and the depth and all, they were not able to achieve that much of a performance or the predictability. In our model, at very elegant system, just 3 properties of the protein were found to be sufficient to predict DNA binding proteins from the others with a high degree of accuracy. So then this is an example that it is not always the deep learning model that works. Sometimes the linear models will work, sometimes very simple features will work. So, and that which model will work in this context will be determined by the combination of your understanding of the models and understanding of the system that you are addressing. So then at the sequence level, if we look at the properties such as amino acid composition, so amino acid composition alone is able to predict DNA binding proteins from others. Again, from a simple linear predictor or linear programming can solve these problems. You do not have to go to very complex models because you have just 20-dimensional space where you can simply use an additive factors, and perhaps in some cases, just 1 or 2 amino acid composite factors can produce high degree of accuracy. In this case, 77% accuracy from sequence alone. So, but then if you go deeper into a question that this is the protein, okay, I know this binds to DNA, but which part of this protein is actually interacting with DNA? I want to know exact amino acid, because in order to design drug or in order to change the binding patterns of the proteins, I want to design very focused the drug, or I need to know the focused target. So we slide a window over a protein sequence, and every protein, every window is then characterized. Then we found that this actually relationship between the sequence on the one hand and the binding pattern of this pattern on the other hand is not straightforward. It is not additive in nature. So linear simple models do not work, and that is where the complexity of the model arises. And then the predictive models that you need here are more complex models, which are based on the machine learning neural networks we used. And then we also moved from the sequence information, putting in the context. Again, we have the sequence is a data obsession, but the evolutionary profile is a data consciousness, that this sequence does not exist alone. This sequence has evolved biologically. So under which biological conditions this sequence has evolved? So evolutionary profile contributes to its predictability. So to put it in the context, because there is a biology behind it, the evolution actually filtered out the sequences which could not interact with DNA and retained the sequences which could interact with DNA. And this information is present in the evolutionary profile. So if we can model this evolutionary profile now, instead of taking the current sequence alone, but also the evolutionary profile, how this sequence has evolved over the generations of other proteins, if we can somehow incorporate that information into our model, our prediction performance drastically improves. And that is how it happened. So in several years ago, we did this multiple models to predict DNA binding sites. And we can see a dramatical improvement in the predictive performance if we incorporate the evolutionary information, and if we go from the linear additive models to the non-linear and non-additive neural network models. So this is an example how the complex models, we keep on doing it, and as the new models arise, new technologies arrive, we try to improve our models. So most recently in 2019, we did this, extended this study further, and we found that, okay, the neural networks were doing fine, but the convolutional neural networks, which can also actually capture relationship between the patterns that exist in unrelated locations. So if these kind of convolutions can also be utilized to predict the DNA binding proteins with a higher accuracy. And that is how we keep on getting new technologies. So on the one hand, the biology grows. So biology grows, new data arises, and new data will prove that your predictive models were not correct, or new kind of systems or new examples of systems come where the model is not working. Then it gives you the feedback. And on the other hand, the technological advances also tell us that these are the pitfalls of a given model. This is where it cannot fit. This is where it can fit. And the non-additive behavior can be modeled with linear regression models. Non-additive behavior can be modeled with neural networks or multi-layer perceptrons. But even the relationships between features can be captured better with, say, the convolutional networks or the models which actually rely on the feature-feature relationships between, say, these patterns. So this worked, and this is another example is for looking at the clusters of conserved residues. Again, this is an example of not overfitting or the finding a prediction, but it is an example of identification of patterns. As I was saying in earlier example, that we may observe patterns which do not exist. So non-additive patterns were detected in the case of conserved residues of conserved clusters of residues. And we found that in the DNA binding proteins, many clusters occur in the conserved. So conserved residues do not just distribute randomly in the space, but if there is a conserved residue in one location, its surrounding regions are also often conserved. And those which occur as clusters of conserved residues, they are more robust, they contribute differently and non-additively to the stability of protein DNA complexes. So these became very popular studies. A lot of people have appreciated these works. And so in this, again, we are using relatively simpler models, not very complex models, and the predictability works very fine. So for instance, I'm not showing it here in this slide, for instance, every arginine residues that occur in the cluster of conserved residue of a given size is in the interface. So I can predict with a very high accuracy that this residue will be in the interface or with the DNA. So these predictabilities we are able to achieve because we have the right kind of domain knowledge going in it. We have the right kind of most appropriate model in the predictive system here. And then another example is the conformational changes in DNA binding proteins. I'll skip this particular example because I have something more interesting to show here. That is the nature of recognition process, what kind of DNA is recognized by DBP. And here we have developed a tool. I'll just introduce the tool here briefly. Two tools I'll mention. One is called the readout, where we have the protein DNA complex. We know that this transcription factor binds this particular motif in this 3D structure of the protein DNA complex. And then from that, can I find new DNA sequences that will also be bound by this protein? And can I find new sequences which bind to this protein more strongly? So this is a very important question for the genome-wide recognition of the transcription factors, and also for designing the drugs, designing the DNA-based drugs, which can actually inhibit a particular interaction or try to mimic a particular interaction in both these cases. So we looked at with this web survey we generated called Readout. This is, the link is not correct here. I'll send the correct link if you are interested later on. So the readout basically threads a large number of genomic sequences onto a protein DNA complex and tries to predict whether a given sequence will sit comfortably in this location or not. So, and the second tool, this worked, and as I said, this could be validated in the biology, in the lab, and in a novel binding pattern, the regulated gene of SAA gene was STAT3 target in SAA gene was found, which were established by our collaborators in Osaka. So this another tool that we developed is when we want to investigate the conformational dynamics of an entire genome, then the tools are abysmally poor. We do not have the predictive systems, and we do not have the first principle technologies. As I said, if I want to do the molecular dynamic simulation from the first principle for the entire genome, I can't do it. Even I can't do it for a small protein or for a small protein DNA complex. It takes long time. And if I want to investigate the entire genome from this perspective, it is almost impossible. So for this purpose, we developed this tool called Dynastic, where we tried to take all the exhaustive set of linear sequences of a short fragments and carried out their molecular dynamic simulation exhaustively for 1,000 odd sequences. And from those 1,000 odd sequences ensembles, we created a dictionary that if this is the sequence, this is the outcome. So this dictionary would therefore be useful for scanning through the genome. For every location, I can find the local conformational dynamics, and these local conformational dynamics can thereby be added up and integrated to find the local regions within the genome which have one type or the other type of dynamics patterns, the conformational ensemble, as we call them. So based on that, we found that many times the sequences look similar, but the conformations are not similar. And therefore, many properties of the transcription factor binding sites could be explained by the use of the Dynastic, this tool. And we were able to detect certain modular regions around the motif, the transcription factor binding motifs, and as in case of STAT3. So more recently, we did this collaboration with Nicoletta Babola in Manchester University to understand the HOX-A2 versus HOX-A3 binding in the case of cranial development of mouse. So this is again, Dynastic was able to predict the outcomes. So there is another, finally, we try to predict the expression, global expression, from the global expression profiles. I want to predict what we call the actual context-based annotation of the DNA binding function. So many times, in our literature, our proteins and genes are annotated, but these annotations are not sufficient to tell us whether a particular function will actually take place in a given context. The context of the protein or gene in which in the context in which this acts is encoded in the form of primarily the gene expression, whereas the functional annotation is based on the sequence or structure. So whether this sequence or this structure, the sequence structure is present in every cell, but this gene or this transcription factor performs this task in this cell, but it doesn't do in another cell. So therefore, the gene expression context becomes important, and that is why our predictions should take into account the biological context, and that is what I called in my slides earlier, the data conscious approaches to prediction. So we developed this multi-platform gene expression matrix data engine called MPGEM, which has hundreds of thousands the expression profiles, and we can create for every protein a global expression profile, how the expression changes under different conditions. And from there, using these global expression profiles, we were able to improve the predictability of the DNA binding proteins. That's to say just briefly. So I think I'll stop here, but there are many things to say and many exciting stories that we come across. And I have given you a glimpse of what can be achieved through these predictions. I'll now let you ask questions instead of me continuing to speak. But before that, I acknowledge all my lab members. I have been very fortunate to have very bright students and very bright team, which keeps growing and which keeps on raising my motivation level. And these are some of them shown here on the screen. And finally, I end up with my personal touch to this in the form of a poem, which says why the place of the predictive modeling. Physics is fundamentalist and fussy. Mathematics is dumb and not yet easy. I told you why physics is fundamentalist, because the first principles are required, and first principles do not really work on large biological systems. And the mathematics is dumb and yet not easy. It is also the question of that exactness, because the data-driven biology and biological data that we collect is not accurate. There's so much of noise, so therefore it is not doesn't work either. On the other hand, the other extreme lies the astrology, which is hasty and crazy, and therefore the human brain uses none of them, because for deep inside its terrain, it knows to represent, test, and train without bothering to explain. But now, as I said, we are in the explainable AI, so this line has to be modified. So data science rules this world, for even if a solution is transient, predictions noisy and incoherent, they work the best for the most because they are intelligent. Thank you.
Thank you, Dr. Shandar, for the amazing lecture. So I would request everyone to type in their questions in the chat box. So I'll just read out the questions from the chat box. So the first question is from Sandra. So she's from European Commission, and she's asking how the biological context-based AI work can help for predicting better treatments of pathogens or chemicals in the environment of food systems, and better predict human individual adversity, since COVID-19 has shown that inter-individual variability and modulating factors can have adverse outcomes in various COVID variants.
Let me understand the question, because it's a bit long phrase. So how the biological context-based AI work can help to be ready. Yeah, so I think the point here, if I understand the question correctly, is that how can we incorporate the biological context? So as you are rightly identifying, the pathogens in the environment, the food systems, the individual differences between the subjects we are treating, and individual differences in their immune responses provide a biological context. So biological context may be personal, and it may be also the context in which the system that we are trying to tweak in operates. So personal may refer to something like personalized medicine, where you need to understand the individual mutations. You have to understand the previous history of exposure of an individual to the infections. So the personal genetic factors and the personal historical factors of the exposure to the pathogens. So this is the biological context that has to be incorporated into the models, in which we are trying to say, okay, this drug works. And if I have all the subjects from one particular area, this drug might work on them, even in the latest stage of clinical trials, or even the early stage of the drug screening. At every stage, the kind of systems I am trying to work on will be influenced by the features or by the biological context which are missing from the model. So if I can somehow incorporate more and more context into my mathematical and computational and data-driven models, I can get to my answer faster.
Right. I think one aspect related to that is also there is like a huge individual to individual variability, right? And when you do AI driven stuff, it's based on huge data sets, which might, combine all the data into one pool. So how much could that predict, inter-individual variability rather than averaging out all the differences and predicting something which could not, apply it on an individual basis? I think.
So if I'm allowed to speak it in a larger context, all theories are meant for the averages, and anybody who is away from averages suffers. So, but in this context, yes, if you have sufficiently large data, so large data, what is the meaning of large data? Large data does not mean that you have thousands of samples. Large data means your data is really representative of the population that you're investigating. Sometimes a data of 500 samples may be more powerful than a data of 5,000 samples. So your data has to be properly representative of all the groups that you are investigating. So if you have this kind of data, and in that data, do you have the annotations for the individual differences, as you are saying? So this data may be completely blind to the individual differences. But when we want to put the biological context into it, we would like to put in this data also what is the T cell repertoire, what is the B cell repertoire, so we can have some idea about the previous exposure, immune exposure of the individual. We can also have the SNPs and the kind of the genetic information about this. We can have current expression profiles. We can have age profiles. We can have the food habits. So all those, many times it is not possible. But if you are still able to, if I say that 90% of my subjects are explained through this, but what about the 10%? Can we take liberty with this 10%? So 10% is not something to be, so that is the difference between data-driven approach, that the 10% that we are leaving out, is it due to some reasons, or it is just because the 10% data is noisy? So that is where the real meaningful differences and meaningful errors and the non-meaningful or the random errors have to be investigated. So many approaches are being developed to make these methods to be interpretable and to be able to incorporate more and more biological context into our models.
So next question is by Parat Sarkar. They're asking, can we predict a best pathway for cancer therapy? As there are so many signaling pathways that are affected in cancer, if you can predict one or two best pathways where a drug can be efficient, then can we overcome the cancer problem? And how can computational model diagnose cancer at an early stage?
So yes, first of all, there's never best, because you would know that very few pathways are actually known. There are thousands and perhaps millions of pathways that exist in human and other living systems which are not even known. So finding the best pathway and how and then the best fine tuning of the pathway. So even if you look at the best pathway resources such as 𝐒𝐤𝐞 and all, they will provide you, broadly speaking, a list of genes which are members of that pathway. They may not exactly always provide you the exact details in which this pathway operates. They may not provide you the fact whether the upregulation of one particular gene in the pathway is helpful or harmful. So just having a list of genes in a particular pathway is, I wouldn't say that it is useless, but this is just the first step. But you like it or not, this is all we have right now. So we can work with the existing pathways and try to find the best solution that we have right now. And then there is a trade-off, whether we can go ahead with the solution that we find or whether we have to still wait. So you have to actually find the trade-off if we administer. So when we, for instance, we had COVID vaccines, there is a debate whether is the drug safe enough? Can we really go for administering it? So here you have to calculate the trade-off. Can we wait for the drug to, can we wait for the virus to invade all of us, even if there are safety risks? So safety on the one side and the urgency of a solution on the other, that will do. And if you really want a solution that will be guaranteed to work, even clinical trials should be carried out over a period of 10 and 20 years, because some of the side effects will not be known until we have already observed them for 10 to 20 years. So that is not practical. So that is the trade-off that we have to make as human beings, as a society. We have to make judgments in each situation.
So I think a kind of question which stems from that is most of biological events, as we know, they are so complicated, right? There are so many molecules, so many processes that are involved in each phenomena, that the more you study, the more you identify more things. So when you are creating a model, so how, at which point do you say, okay, I have to stop? Because, so when you're incorporating elements into it, so at one point you have all the necessary elements. Beyond it, it becomes too complex. If you have lesser than the requisite number of elements, then it is too simplistic. So how do you decide, these are the kind of elements that I want to incorporate into my model when you're building a data-driven model? I think that's, so how do you kind of decide that point?
So, well, there are safety standards. This is not my area where we choose a standard. There are standards when we say we declare a drug, for example, to be safe or not, and whether we are ready to go to the next stage. There are standards, and so there are multiple stages of drug. If you're looking at drug discovery aspect, so there are multiple stages of drug discovery, and there are established practices. And the experts, there are experts who totally focus on one aspect of this. Okay, how can we go from this stage to the next stage? People spend their entire life in just answering this single question. So as kind of superficial experts in this field, what we have to do is to really look at their work, find their work, and borrow the right cutoff. So I would not be in a position to recommend an exact cutoff, but general principles would be, of course, that you have to always look at the trade-offs and the costs, not just in the monetary cost, but also the social cost, the individual cost. So that have to be worked out. And this is a matter of rigorous research that people do.
So Advait Bhagwat is asking, given partial data, can you predict unknown steps in a pathway using data science?
Yes, people are trying to do this. For instance, you might try to find out, okay, if there are multiple pathways. Even pathway is not a single entity. Each pathways has sub pathways, and one version of pathway is activated in one condition and another version in another condition. You may have TLR pathway, for example, toll-like receptor pathway, but this pathway may act differently if mid-88 gene is expressed or not expressed. So if adapter changes, the pathway acts differently. So this is not a monolithic problem. Yes, but people do try, and some pathways can be modified and can be you can create artificial set of genes. You can go beyond pathways. You can also look for gene ontology enrichments. You can go for gene enrichment analysis, which are not totally dependent on pathways. Gene set enrichment. So many gene sets have been put together, which are not exactly in the form of pathways, which may work as a kind of cascade or which may work independently. So many different alternatives to pathway analysis also exist.
Bharat is asking, could you please comment on application of AI in predictive toxicology, selection of data sets and tools for predictive toxicology?
Yeah, so this is an area in which a lot of money has been spent, and there's a very active area of research. So people are trying to correlate, for example, the whole slide image data with gene expression data and metabolic data. So the integrated approaches are being developed. For instance, earlier I was working in this National Institute of Biomedical Innovation in Osaka. So they developed a whole system, what called TG-GATE. So that TG-GATE is concurrently they collect the histopathological images, and for each image they also collect the RNA-seq expression data. So relating the toxicological impact on the images histological at the histological level, and trying to connect it to the gene expression level is one such very big issue. But again, it is so challenging because the kind of images that you need to collect, the kind of tissues that you collect are huge. Even images they have collected is several gigabytes single image. And the expression profiles in that data last time I saw is about 300,000 expression profiles. So just putting the data into memory and developing predictive models is a challenging task. And that is where the inputs from the technical and computer scientists are going and are required.
Sandra, I think, thank you for mentioning the concept of calculating the trade-offs, which is the issue in science for policy decision making, and a wonderful session. So just one last point, which I was thinking, like for example, during data collection itself, right? Sometimes the data collection process it can be biased. So how do we account for that when you're building AI-based models? Because that has been found, right? Because when AI-based models were created, it was in some cases biased because the initial data collection was biased. So is there any way to kind of validate the data itself and checking for biases? And
yeah, in the field of perception, there is the actual perception of objects by individuals. Psychologists have known this for a very long time, and they say that we see what we want to see. So that is just an example of the kind of biases that may appear, but this is not at the collection stage. This will appear everywhere. because we have been funded a project, we are forced, and we are trying to always look for this drug should work. And if we have said something in the past, so these are other aspects, but your question, scientific question of how to ensure that our data are not biased. So again, first we set up our goals, that whether we want to design a drug, are we designing this drug to work in our population, or are we going to design this drug for the entire world? Are we going to design this work to work on one ethnicity versus the other, or we want this to work universally? And you cannot rule out, this is the law of nature, that the drug that you design will be biased, may work on certain individuals and will not work on certain other individuals. So first we set up our goals, which are unbiased, and which is again a matter of policy, as we were just mentioning. So we make these goals, and after you have set up these goals, now you collect the data appropriately. So at the time of collecting this data, so there's a whole branch in statistics called the experimental design. So people find robust statistical methods to design their experiment, so that the questions that they have framed are appropriately reflected in the data they collect and in the hypothesis they derive.
So if I don't see any more questions in the chat box, so okay, there is one comment by Bharat who was saying, could you please provide some insights on recommended public domain data sets?
Yeah, so this is a very important issue in our research, and data sharing, data privacy. On the one hand, we want all the data to be in the public domain. If we want the research to progress, every data should be in the public domain. On the other hand, it leads to the misuse of this data. So we are always in conflict, in two different conflicting values, one of the privacy and other of the availability. So when data availability speeds up the research, but data availability also exposes us to have, say, for example, bioweapons, which will only work on us in the extreme case. And you can have, of course, biological weapons, which will work on one particular group of people. So that is why identification or providing this data poses real challenges. It is not just a matter of making the data available. So there is always a very serious question whether the data should be made private or should be made available. Now, what is available in the public domain? That is another question. That in public domain, if you look, particularly biomedical data, very little data is available. And most of the times, the best papers come from people who have exclusive access to the data that they are analyzing. If you go around the best research publications, even the basic science people, they are keeping their data opaque. So their strength in their publication is not really their science. Many research papers, many big research papers are coming from big labs. So what is the strength of those papers? The science that they're using, the science that they can actually generate out of this data is not really that great. Perhaps the same science can be done in small villages anywhere in the world. But they have the advantage because they have the access to the high quality data, which has been collected with at a very high cost. So for instance, if you look at some of the big projects of the cancer genome and other projects, and the image data, hospital data, you will find lots of papers which only the people who publish that paper, only they have the access to data. Other people cannot even verify whether the findings that they are reporting are correct or not. And that is why some controversies arise. Some controversies did arise in the case of COVID drugs, as you would remember that recently it happened. So access to data is a critical issue. And since I am happy that some of the policy experts are here, and this is a very important question from the science point of view and also from the social perspective. Researchers would want that every data should be available to them, but there are huge implications to this, and some kind of balancing. Availability there may be some kind of agreements between the providers. Many times the data is generated by public money. A lot of data in hospitals is generating by my money, but I don't have the access to the data. I cannot verify what the people are publishing. Only selected scientists are publishing the data. So we may not even know whether what you're saying is honest, whether they have thrown out some of the data that did not suit their models. So this is a problem, and I think this is a serious problem we have to consider.
I think that is particularly true of like data from Indian population, right? Because many of the public accessible database, they mostly don't have population from Indian population. For example, there's something which like many of the people I spoke to, they also said like when even the data sets that are in the public domain, mostly they don't have data from Indian population.
Yeah, so here, Surat, this is there are two reasons for this. One is of course, privacy and that issue we discussed. The other issue is that, first of all, our clinical community, medical community is not very good at collecting, has not paid attention to collecting the data. And the quality of data they collect is not really that much. So if you look at these, for instance, the website 𝐒𝐤𝐞𝐵, so you will find lots of data for different diseases, for different images, COVID, pneumonia, and malaria, everything. So they have collected the data, and the data has been collected under controlled conditions. Our experts have focused on clinical information, our clinical practices, but there has not been a very robust system of collecting this data. So, and wherever the data exists for the Indian patients, for the Indian subjects, the quality of data is not good. You'll find lots of errors. The resolution of the images is not good. So this is changing. I mean, I would not say that this is a problem that we have to just give up on it. This scenario is changing. More and more data is getting collected, but still we are not there where we can actually utilize this data for developing India-specific solutions.
So Sandra is asking, is there, can we identify the various different kinds of biases such as experimental bias, information bias, selection bias, method bias, expert bias, design bias? Can we differentiate between these different kinds of biases in our data?
Yeah, this is interesting. Actually, a good data scientist can know that this data is biased. The data actually speaks about the truth of itself. So there are data techniques where we can tell that this data has been manipulated or this data is wrong and this data is biased. So there are, for instance, we can do clustering. We can see how many clusters exist where the data are biased and what are the observations that are impossible to have. So there are data validation techniques that can pinpoint about the data biases. But if you do not have access to the global scenario and you have data collected from a very biased sample, so what we can demand at the time of dealing with data is we can demand the complete details under which this data was collected. So how many people were administered a drug and at what was their age group and complete details. Many times these details are not available. So if you, the enforcement agencies may make it more important for the data providers to identify that information.
We'll just take one last question, which is from Bharat. They're asking, can we bridge the gap between in vitro and clinical findings using AI?
Yes, of course, this is already happening. So there are multiple stages. As I said, you will start with in silico, then you go in vitro, and then you go to clinical. And within the animal models, what you observe on mice may not be observed on human. So there are all these different stages, and of course AI is helping it. For example, I remember that there was recently a competition called Dream Challenge, organized by IBM and other companies, and this was to predict the human gene expression from the availability of the mouse gene expression, phosphorylation and gene expression. So yes, at every stage when you go from one to the other, starting from in silico to the final clinical approval, there are multiple stages where information is lost, and the AI is trying to bridge that gap and trying to decrease the rejection rate at the later stage.
So I think at this point, one last question we'll take from, they're asking, for building ML models, where do we learn the models from? Is there any specific database?
Where do we learn the models from means which model to choose? Is that the question?
For building, I'm guessing, yeah. Or
yeah, so I think this question is not clear. Probably you want to know which machine learning model to choose in a given condition. And our lab is actually working on these questions. We have published a couple of papers on how to make the best choice of a model before we actually use the model. So one thing I pointed out in my earlier discussion today was that many times we keep on trying different models because there are so many models and there are so many ways to represent your data. One of these models will work. So if one of these models works, does not mean that this model really is meaningful. So if you try too many models and one does not work, then you end up having an overfitted model and a model that works only on your data. It doesn't work on other people's data. So there are techniques of cross-validation, trying to have a, mask the data and keep it as a holdout data. But these are not honestly used by researchers, because when they report it finally in the research publication, then they do have a holdout data, but they have tried all the models, and the model that worked best for the holdout data they report. So this was not really a holdout data in that case, right? Because that has been somehow used in deciding which model to use. So the best is look at your data and ensure that what kind of just assess whether the data is complex, whether a simpler model will work. Start with simple model and then introduce complexity gradually. This is one approach. And another approach is that you use the state-of-the-art model today. That is what many people do. But my personal preference would be to start with simpler models and introduce complexity into the model gradually and see how the model is improving, so that you can monitor how the performance is improving as the complexity increases. And then there are techniques to ensure that the model does not get overfitted because of the use of the multiple hypotheses or because of the use of too many features.
Okay, with this, we'll stop. And thank you, Dr. Shandar, for the amazing both lecture and post lecture session of Q&A. So one last thing I had mentioned in my initial thing, that we have a quarterly newsletter. So I've just posted the registration form for the newsletter in the chat box. So please register if you haven't registered for our newsletter yet. And thanks again for both our speaker and all our participants who joined us on a Saturday morning for an engaging session. And thanks everyone, and hope to see you at our monthly session next month. So thanks again, Dr. Shandar, for the session, and we'll be putting up the recording of the session on our YouTube channel soon. So yeah, thanks a lot.
Thank you.